Alright, the topic today, we are going to transition into cell cycle and reproduction. And try to answer the question, how do cells reproduce and grow? So, before we get into any particulars, why do cells divide? Well, it depends on what type of organism we are talking about. If it is a multicellular organism, there's a whole bunch of cells in that organism. The purpose of cell division is to increase in cellular number. Now, the reason why you'd want to increase in cellular number is for overall growth. If you compare yourself now compared to 10 years ago, you've probably gotten taller, you've probably gained more weight. The reason why you are bigger is because you have more cells. Repair, if you've ever had a cut or a break, how your body goes through recoveries is it fills in the gaps with new cells to kind of keep things whole again. And then for renewal, this is going to be for basic overall maintenance. Cells have a lifespan. When some cells start to kind of break down, they need to be replaced. And they're replaced through cell division. On the other hand, if you are a unicellular organism, you only have one cell. That is it. One cell does all. You're not getting any more. So this is a form of reproduction. Instead of increasing cells, we're increasing in organisms as a whole. So this is a form of asexual reproduction because there is only one parent involved. So we start with one organism and we end up with two organisms. Now, from a prokaryotic standpoint, remember, prokaryotes do not have a nucleus, so they cannot undergo what's called mitosis, which is defined as the division of the nucleus. Instead, they go through something called binary fission, which is going to be similar. We're going to talk about mitosis here in a couple minutes. Most importantly, when does cell division happen? And it happens when cells get too big. Remember our surface area to volume ratio. Volume increases faster than surface area. So when a cell gets too large, it is no longer efficient to kind of get things quickly into and out of the cell. So it hits the trigger that is now time to divide and get smaller again. So basic cell cycle. Majority of the cell cycle revolves around a stage called interphase. It has three different phases to it. About 78% of the cell's life is going through interphase. This is basically prepping for cell division. If there's anything to remember about interphase, is you are making a copy of everything. Making a copy of your organelles, replicating DNA to make sure we have two sets, and with this leads to growth. Because you have to accommodate for twice as much stuff. You got to get bigger. But at some point, getting too big is a problem, and that's when you're going to prep for mitosis, the actual division of the cell. Which, remember, mitosis is defined as the division of the nucleus. So when we look at the cell cycle, we have a couple things that we need to break down in terms of what is happening at each step. Now, interphase. Remember, this is 78% of the cell's life. It is the prep step. The first phase in interphase is called gap 1, or simply G1. Here, we're going to see growth. And we are going to see the replication of organelles. So we're going to have twice as many. The big organelle that we're going to replicate that's going to be a big helper with the cell cycle is the centriole. The second stage in interphase is called synthesis, or the S phase. The only thing happening here is we are copying DNA, making sure we have two sets of it. And then the third stage is called GAP2, or the G2 stage. And what we see here is more growth and basically prepare for mitosis because we've gotten too big. Now, mitosis, within the cell cycle, you'll hear referred to as the M phase. This takes up the remainder of the cell's life, going through the actual division process. And remember, in cell division, whenever we have mitosis, we are dividing the nucleus, and the only type of organism that goes through mitosis is going to be your eukaryote. Eukaryotic cell because it has a nucleus. Prokaryotes do not undergo mitosis. Prokaryotes just do a quick. Prokaryotes, no mitosis. Instead, they do something similar called binary fission. Now, an important thing, cells are all different, and they're going to divide at different rates. So, this is going to vary widely. It's going to be linked to the body's need. And the length of gap 1 in interphase varies among your cell types, as we see down here with the approximate lifespan of your cells. Skin cells have a very short lifespan because there's a lot of wear and tear with your skin being the exterior barrier of your body. So we go through skin cells very fast, whereas other cells may take several days, several months, or several years until they need to go through the renewal process and be replaced. Now, cell size. We've talked about, remember that surface area to volume ratio. We want it to be high. So we want more surface than space inside. So that smaller cube is kind of the bread and butter here. So what happens if it's not a high ratio? Well, if it's too small, then there's not enough space. Remember, we are making 2 times the amount of everything. So you need space to hold all that in. If it's too large, then it's too slow to move things in and out. It's not efficient. So cell growth and division must be coordinated. It's like Goldilocks and the Three Bears. You need it just right to work out. Now, when we get to genetic information, DNA is where we have our genetic information. When it is not dividing, we call it chromatin. And it kind of looks like spaghetti. It's kind of in a loose state. When it is dividing, it condenses into chromosomes. Why? Because it is easier to move, and it is less likely to be damaged. Now, as we look down here, the chromatin looks like a bowl of spaghetti. Whenever we are getting ready to divide, we need to condense the chromatin down into a better usable form, and that's when we get into our chromosomes. We have two different chromosomes right here, two copies. Remember, we do copy the DNA, so two of the exact copy forms that X shape. Now, within a body cell, we have 46 chromosomes, 23 pairs. One set from mom, one set from dad. And that 23rd pair determines what your gender is. XY is going to be a boy. XX is going to be a girl. And this is in every single one of your body cells. And we're going to define that as any cell that is not sperm or egg. So now we're getting into the actual division part, which is mitosis. Mitosis, think PMAT. Prophase, metaphase, anaphase, telophase. Now with our chromosome structure, we need to first identify that a chromosome is one long continuous thread of DNA. DNA wraps around proteins, and those proteins are called histones. And this is going to form chromatin. And then remember, when dividing, your chromatin condenses into chromosomes. And these chromosomes have an X shape because in interphase, we have copied DNA. So you have two of the exact same chromosome, one on one side, one on the other side, and that's why we call it a duplicated chromosome. Each side is its own individual chromosome, but they're the exact same thing. Each side of the X is called a chromatid. Now, in the cell cycle, we're going to go back to interphase real quick, just to do a quick review. So everything starts off with interphase, majority of the cell's life. What's happening here is DNA is copied. We see growth, cells getting bigger, and you see duplication of organelles. So we're getting bigger, and we have 2 times the amount of everything. Once we get too big, it's going to trigger mitosis to happen. The first step in mitosis is prophase. Now in prophase, chromosomes condense, nucleus dissolves, spindle fibers form. So what we see here is your nucleus starting to break down. instead of that kind of thing of spaghetti, we see it condensed into chromosomes. So one side is one chromosome, and the other side is the copy that was made. Now, when we talk about prophase, I like to think P for prep. This is a prep stage. We're preparing. After prophase, we get into metaphase. In metaphase, chromosomes line up in the middle. So we see that right here. Chromosomes being lined up in the middle. Now how this happens is you have these spindle fibers that are connected to your centriole. So finally, we're talking about your centriole in terms of what they actually do. And your centrioles are in pairs. So one pair on one side, one pair on the other side. And they almost act like puppeteers because we have a component of the duplicated chromosome called the centromere, and your spindle fibers are connecting to the centromere on either side. So your centrioles are kind of putting the chromosomes in place where they need to be. With metaphase, M for middle. Next, we have anaphase. And in anaphase, chromatids pulled to opposite sides. We are separating out your duplicated chromosomes and having one full set on one side and one full set on another side. So it's almost like we have this no man's land that's kind of between them, and that's where the cell is eventually going to split. So with anaphase, I like to think A for away, because the two sets are being pulled away from each other. That X is being pulled apart. After anaphase, we have telophase. In telophase, we have new nuclei form and chromosomes uncoil. So we start to see new nuclei form. Remember, mitosis is defined as the division of the nucleus. So one nucleus ends up becoming two. And we have a full component of DNA in both parts of the cell as we start to see the separation. You also have a full set of your organelles, since we do have our two centrioles on one side and our two centrioles on the other side. Because in interphase, we made twice as many. Our final step is called, and telophase, T, I like to think 2, because now we start to see two cells start to form. Our final one is called cytokinesis, and this is simply just the division of the cytoplasm. This is going to be the separation into making them finally two cells. Now, overall, since there's a lot going with prophase, this is the longest step of mitosis. And anaphase, this is the shortest step of mitosis. Now, animal cells and plant cells do have a slight difference when it comes to cytokinesis. Animal cells simply pinch in. They have what's called a cleavage furrow, where that no man's land formed during interphase simply pinches in until we have two separate cells. Plant cells can't do that. They form what's called a cell plate, which are fused vesicles. So because of the cell wall, it can't pinch in. So these vesicles start to build up and eventually form a new cell wall. Okay, that is it. Thanks for watching. Let me know if you have any questions.